Hello, everyone. Welcome to today's investigation. I'm Shannon Reeves, and you should be in section 39 or section 40. This is Leo, and he's asleep. And this is Ravioli, and she's happy. All right, let's get started. So, just some announcements to let you know. I should have provided some comments for the first couple of lab summaries. I won't be doing it as much anymore. I will be filling in the rubrics so that you know where you lost points, but I'm not going to leave as many comments. But those who are missing a couple of points, usually it's because you're missing parts of the rubric. So be sure and have the rubric out while you're writing, as well as probably how to write a lab summary and how to properly cite. And this week we'll have the statistics portion of the resources available for you in the actual module because you'll be using statistics again. We'll also go a little bit over the results section. We've been going over each section to make sure that you know what content is needed for it, and we'll go over the results section this week. So this week you'll complete a research proposal just like you did last time with the 5 elements. You have your hypothesis, your independent variable, dependent variable, your control groups identified, and your experimental groups identified. You will carry out your experimental design, which means watching videos that we've provided for you to collect the data and make graphs. You'll complete a lab summary, which has a graph and reports your statistical tests, and upload that to Canvas along with a Flipgrid video for this week. So this week's general question is, why are some heart rates different than others? Again, you'll come up with your own question that is generated from what you want to do with your variables that has a little bit more specific question to your hypothesis than this one. But we'll be talking about action potentials. So how is heart rate regulated? As you know by now with lecture, an electrical signal traveling down the axon or over the synapse is called an action potential. So those are affected in lots of different ways. You're probably more familiar with this picture from lecture, but you should know by now that neurotransmitters are the major players that send signals across a synaptic cleft, and it reaches to its final destination either at another neuron or a muscle that makes that muscle contract. So we talk about seizures and paralysis. So you'll either increase your action potentials to potentially cause a seizure or block action potentials, which can cause paralysis. So the neurotransmitter that we've been talking about is acetylcholine, but what happens whenever you try adding different substances, different chemicals to the equation, and how does that affect heart rate? So the drugs that we have today may either mimic, which would increase your action potentials, they'll block it, which would stop action potentials, or have no effect at all on action potentials. So the chemicals that you will be given, you'll have to decide which two do you want to compare and what do you think might happen to the heart rate of a Daphnia, not a human. But we do know what happens to humans, so you could use that as a causal statement for your hypothesis. The first drug that we have for you is nicotine, that we know is a stimulant, ethanol, which is a depressant, and caffeine, which is another stimulant. So you can hypothesize in comparing 2 of these groups, whether you want to compare the 2 stimulants together, or if you want to compare one of the stimulants to a depressant. So this is a picture of the chemicals that you'll be able to use. But first, let's talk a little bit about the Daphnia and what we need to be looking at. So your Daphnia sits like this, and it will be moving down here a whole lot. The students think that these often are the heart because they're moving. It's not. It's gills. They're kind of like legs. So they kind of just move around down here. Don't be tricked by it. That's not it. The other thing that students often think that are the heart rate is this right here, E. They think often that it's circular and it's oval shaped and it's pretty close to the heart, but it is not the heart. Those are babies. So don't accidentally count babies. They don't move and it'll look like your Daphnia is dead. So B right here is an eye spot. That's where they see. You have little antenna that poke out right here. And D, the most translucent part of the body is the heart. So this body being translucent is a really cool way that we can experiment and actually see what the heart is doing. We couldn't do this with like a tiger or a human, obviously, but we can do it with these little crustacean like Daphnia. So you can see through the heart and you can see through the body to see the heart. It's pretty evident in the videos. You can see it. Just make sure that you're counting the correct thing and not accidentally counting legs. So we'll follow the lab background. And in the background, you should have figured out that we are going to do two different values in this. We're going to find two different values. The first one is our before or pre-heart rate before adding the chemical. And then we'll wait 10 minutes, wait for the chemical to set in, follow the protocol, which you won't do that. We have done that in the videos that we provide you with. You won't be doing that, but you need to know about that because you'll definitely want to write about that in the methods. A lot of students have been confused about what needs to be in the methods as far as protocol is concerned, because obviously we don't need you to tell us that you've turned on the microscope. Or you're breathing in and out while you're doing the experiment, right? But we do need you to tell us how much of a substance was used. Generally, how long did you have to wait between steps? What were the steps? Did you use chambers? Did you put, Daphnia on a slide, or did you put it in a petri dish? These are small things that are helpful for your reader to know how the experiment was performed. So then after we do that, after we wait the 10 minutes, we record the same Daphnia's heart rate after adding the chemical. So we'll talk about in a couple slides why it is that we're using pre and post as control and experimental group. But first, let's talk about tips for analyzing this data. So a lot of students are still leaving out how data was analyzed in their reports. And the first couple of labs that we didn't have a whole lot of analyzation to them, so that's probably why it's confusing. But anything you do to the raw data is considered analyzation. If you average it, you're analyzing the data. If you're doing some sort of calculation in order to find a different number, like a rate, then that is analyzation. So in the cellular respiration and in the photosynthesis labs, if you didn't mention how you got rate and that is truly what you were getting, not the overall CO2 production, then you probably missed points. So we're not getting the overall heart rate. That is your raw data. So how data was collected, you'll say that you looked under the microscope and counted the heart rates within 10 seconds. But that's not analyzing the data. How it was analyzed is right here. Remember to calculate beats per minute. So we didn't give you a minute-long video. We gave you 10 seconds in real time. So that is technically your beats per 10 seconds, not your beats per minute. So you will have to take that value and multiply it by 6 because you don't have beats per minute. So that's one thing that you'll want to mention. To compare the effects of one chemical to another, you'll need to determine how much of an effect each chemical had on the heart rate. And that's really what we're going to be doing, is finding the percent difference. So we've provided an Excel file for you that actually makes this calculation for you, but I'll give you the calculation so that you can write about it in your methods. Essentially, what we want to do is compare how much your heart rate changed when given one chemical, how much your heart rate changed when given a different chemical, and see if those two changes are similar or different. So that's really what you're comparing. So why does each Daphnia serve as its own control? The pre-heart rate is going to be your actual control group. And that's because each heart is a little bit different in each Daphnia. And you want to be able to see how much has changed when it's in the presence of this chemical. So for an example, I have this weight loss surgery because it has similar themes. So if I were to do a weight loss experiment and I had a positive control group of diet and exercise, we know that works, so that's a control. And then I had a negative control of doing nothing. And I had this brand new weight loss pill that I thought really helped people lose weight. And that would be my experimental group if I was doing a randomized clinical trial. So my independent variable would be the weight loss strategy, because we have diet and exercise as a strategy. We have nothing as a strategy, and we have the new pill as a strategy. And I would be wanting to find the percent difference in weight loss. So how much did each of those strategies make me lose weight? So that is the percent difference that we're calculating. It's the pre-weight minus the post-weight over the time experiment. So we're going to find the percent difference that we need between each chemical. So before we go on into the rest of what's needed, let's really quick visit the results section. So we're going to revisit the experiment with Ravioli, where she was finding different places to visit or to nap around the house. And we have the orange chair right here, which is that guy that Leo's stealing, is 68 degrees. A windowsill is around on average 72 degrees. And an ottoman is around 69.5 degrees. And a cat bed, our positive control, because we know that Ravioli likes to hang out there, is 74 degrees. So whenever I report my results, some students are still having some trouble in figuring out what really do we report. Before I go into what you do report, what you do not report is how this data looks in comparison to your hypothesis. Don't talk about your hypothesis yet. Save that for the conclusion. That's where you want to talk about that. Results is just for reporting results. That's it. So you're going to want to report values and you're going to want to report trends. So the rubric says main results and trends in data are fully and correctly described in the text. So if you do report values and trends, but you report it wrong, you'll get a point off. But if you just do maybe the main results and no trends, then you'll get a point off if it's correct. We need main results, which means the actual values for each of the groups that is listed right here and how they compare to each other. So one sentence giving specific values of what you measured in your experiment, and then one sentence describing the overall trends amongst the groups. So I put for this, on average, because that's my calculation, right? Let's see if I can do that again here. So average is what we are doing. On average, the orange chair spot was 68 degrees, the cat bed was 74 degrees, the ottoman was 69.5 degrees, and the windowsill was 72 degrees. I'm giving the exact values that I actually calculated. Overall, we can see that preferred areas have higher temperatures than other areas of the house. Overall, the groups that she really likes, like the cat bed and the windowsill that we already know she likes, those did better. So in your photosynthesis experiment, you should have given me exact values for what we were calculating and telling me, well, generally in these colors of wavelengths, we had more plant growth than in these, or this one generally did better in this way than this one. So make sure you're comparing those things. And before I forget, in conclusion, you'll want to make sure and connect the dots at the very end between your independent and dependent variables. So whenever it asks what was learned, we don't necessarily mean like, hey, pop trivia, what did you end up learning that you didn't know before? It's more like, no, what is the relationship between the groups that you did your experiment on and the dependent variable. So in photosynthesis, you should have given some small explanation about how wavelength or light color changes the amount of plant growth that's being experienced because of photosynthesis or because of pigments. So you need to tie in those loose ends right there about what your independent variable and how your independent variable affects your dependent variable. So now let's talk about statistics. So you'll need to follow a flow chart in order to find out what your statistical test is that you'll need for this. So just as a reminder, you probably won't be using the same statistical test that you used previously. So you'll want to follow that flow chart to figure it out. But by doing that, you need to figure out what your independent and dependent variables are, whether it's nominal or measurement data. So I call those categorical and continuous data. Is it data from normal distribution? So you'll do that normality test that you were supposed to do before getting your p-value. And then figure out what type of test that you actually are going to use to get your p-value. and then report that using the template from section R5 of the manual, and that'll be reported in your results section. So you're trying to find a p-value that will be comparing your two groups, and that essentially means, is the difference that you see between these two chemicals that you've added legitimate, or is it just chance that it ended up doing that? So your p-value, if it's low enough, it says, no, it's not chance. This is actually real evidence, versus, no, it doesn't really look that significant. And remember, saying something is significant is actually a scientific value. It's a measurement. So don't use that term casually whenever you're reporting your data and your results. So this is that flow chart I was talking to you about you'll use, and this is going to be available for you in this week's module it's called R5. It's one of the references, along with how to properly cite that we've given you this week. So you'll follow this through. You first need to figure out what type of data, whether it was nominal, all nominal, nominal and measurement, which is most stuff, and or if it was all measurement. Then you follow that by finding out what how many groups you had in your experiment. If you had 3 or more, which was the case for the cellular respiration test, then you should have done one of these 3 tests. And if you have 2 groups that you're comparing, then you would do one of these 4 tests. If it's a paired sample, then you would go over here. We will not have paired samples this week for our data. I'm not going to go into the explanation of what a paired sample is, because it might actually confuse you, because we do have some data that seems like it's going to be paired, but we won't be using that. So you'll use the chart to figure out where you need to go here. And again, that will be available for you on Canvas. The second thing that we need you to do is to continue to paraphrase, and just so that students know, paraphrasing means that you put this somewhere in your report. So this is not something that's added at the end or its own little section. You either put that in the introduction as helpful information that tells you about your hypothesis. And helpful information doesn't mean like telling us about, so let's see. This paraphrase talks about pacemakers. If you talk about pacemakers, it's totally irrelevant. Doesn't matter. What we really want to know is what is relevant in this particular paraphrase to your experiment. So a lot of students used the supermarket and how they want fresh vegetables and fruits for their enzyme lab summary. And really, that's not helpful for the reader, because it talks about a situation that isn't super relevant. Instead, there was a lot of good information there that talked about why enzymes cause browning and stuff like that that was more interesting. So make sure you're taking the gulp of information from this that isn't irrelevant, the stuff that's really helping either frame your hypothesis or support it in the conclusion. If you end up getting results that say, hey, your hypothesis is supported, you might use some of this to say, look, some other smart people agree with me. Or if your hypothesis isn't supported, you could use this information as, hey, look, this is what we found out in the end. This is really what determines heart rate. So keep in mind that for this experiment, you will want to figure out why it is that these chemicals may be having an effect or no effect on the Daphnia, and that will be what was learned in your conclusion. So here's the information for your journal so that you know how to cite at the end of your literature cited. And again, Dr. Shaw has put on the how to properly cite document in this week's module. So take a look at that if you're unsure, because we still have a lot of people getting half credit for that. So again, you'll complete a research proposal form, you'll carry out your experimental design, collect data, make graphs, make a Flipgrid video, report all that stuff in a summary, and turn it into Canvas by Friday at 5 p.m. as always. And just as a reminder, you're welcome to send me an email and ask, but I won't even respond to it. No late work, no partial work, no credit for kind of getting things right. So everything is due Friday, 5 p.m. indiscriminately. Everybody gets the same opportunity. But keep in mind, I'm here. If you have any questions or are confused by the assignment, you can always email me or set up time to meet with me in office hours. So that is it for this week. I hope you guys have a safe and healthy week. Bye.